A warm good afternoon to one and all. So first of all, I thank the OMICS group for giving me such a wonderful opportunity. So basically, I'm Jency Blesson from India, and I'm working in the field of antibiotic resistance in bacteria. Maybe a very different field and the topics on which you people talked. So this is synergism between antibiotics and plant extracts against methicillin-resistant Staphylococcus aureus. So my journey started from India, and you can see the southernmost part of India. It's called Kerala, from where I hail from. And these are some of the geographical things which you can see over there. A very beautiful place. And this is my esteemed institution, Sree College of Engineering. The same thing. And coming to the introduction. So the infectious diseases are caused by both bacteria, fungi and all. And why this antibiotic resistance? You know that nowadays so many people, that is nearly 50,000 people die each year throughout the world due to infectious diseases. And many of the Asian countries, you can find lots and lots of antibiotic resistance. So the development and spread of resistant microbes to currently available antibiotic is a worldwide concern. So to the the fact is, nowadays when we go for single drug resistance, it is not seen. Most of the times it is multi-drug resistant. So in some of the countries, at least in some of the countries, they are going for medical audits, including nosocomial infections, and also for the personnel who work in hospitals. So they rarely, they very clearly check for the presence of the antibiotic resistant microbes in the people who work in hospitals and other institutes. So the pace of development of, and that is this next concern, the pace of development of new antimicrobial agent has slowed down while the prevalence of resistance has grown at an astronomical rate. The rate of emergence of antibiotic-resistant bacteria is not matched by the rate of development of new antibiotics to combat them. So, these are the common bacterial targets right now we are using. That is, again, cell wall synthesis, cell membrane. These are the targets you generally know. Protein synthesis, inhibitors, and all that. Why drug resistance? So these are the major reasons why the bacteria is resistant. And one more reason is there, that is resistance plasmid, which is seen in the bacteria. So one is efflux pump, the decreased entry, altered target site, bypass pathway, and enzymatic degradation. So these are the ways by which bacteria gains resistance to drugs. So how to meet the current scenario? So you have the three options. One is that using the current antibiotics, because very rarely the pipeline, like the introduction of or the synthesis of antibiotics is very rare in the pipeline, antibiotics. So you can go for the natural plant extract, and the third one you can go for the combination therapy or synergism, the area which I work. So the first one, as you know, I'm not speaking on that, the current antibiotics use. The second one is natural plant extracts. So what is the drawback of natural plant extracts? In many of the cases, due to the geographical location and other reasons, the natural plant extracts is being used, but it is not as efficient as the current treatment protocols. So that is why I have selected the combination therapy. That is the strategy employed to overcome these resistant mechanisms in use of combination therapy. So this combination therapy can be of different plant extracts or plant extracts with standard antibiotics or antibiotics with some chemicals. The association of antibiotics with plant extracts against resistant bacteria will have a different mechanism of action, and it may lead to new choices for the treatment of infectious diseases. So why combination therapy? It is to expand the antimicrobial spectrum, to prevent the emergence of resistant mutants, to minimize toxicity, thereby exhibiting antimicrobial activity greater than that would be expected from each antimicrobial drug individually. So synergism. What is synergism? It is a positive interaction created when two agents are combined and together they exert an inhibitory effect that is greater than the sum of the individual effect. So the advantages are increased efficiency, reduction of undesirable effects, increase in stability or bioavailability of the free agents, obtaining an adequate therapeutic effect with relatively small doses when compared with a synthetic medication. So these are the studies. I think the first paper was published in the year 2008 in the area of synergism. And a study was done in Mangifera indica, and only very rare plants are taken for this synergistic study. What is the procedure that we employ? So the normal procedure is, first we need to identify the microbe, whether you want to work with methicillin resistant bacteria or any other bacteria. Then selection of plants, extraction of plant leaves, preparation of the mixture, study of the antimicrobial activity of the mixture. Then separation of compounds, then preparation of a synergistic drug, other toxicity studies of the mixture, etc. First one, selection of bacteria. The bacteria which I have selected is methicillin resistant Staphylococcus aureus. So the characterization, because it is already available in our lab, we are working on we are working with several bacteria like this. So I have retrieved it from my same laboratory, but just for the characterization I have done the basic test. Then these are the plants which I have chosen. The first one is Mimosa Pudica, commonly known as a shame plant. It may be available, like you may be familiar with that, and it belongs to a Tracheophyta family. So this is the classification details. Second one is Ixora coccinea. This is another plant called scarlet jungle flame, which is very common in India. And Colocasia esculenta, this is also very common in our location. This is called cocoyam. The fourth one is Boerhaavia diffusa, and it is called red spiderling. And these are the antibiotics which we have chosen: nufloxacin, gentamicin, ampicillin, erythromycin, ciprofloxacin, and oxfloxacin. So the antibiotics are chosen based on, I have chosen this for the use that is, for the current use, that is, it is not used against resistant Staphylococcus aureus. It is the ones which are used against staphylococcal infections by the doctors. So based on that, I have chosen the antibiotics. So the extraction method, we have followed the Soxlet extraction. This is an apparatus which is commonly used for the extraction procedures. And the study of antimicrobial activity was done by the diffusion, the very common Kirby-Boyer method, UV spectrophotometry, and minimal inhibitory concentration, or MIC. What is MIC? MIC is the lowest concentration of antimicrobial agent in combination at which visible bacterial growth was inhibited. And this is a protocol for that. That is, Muller-Hindenburg was chosen for that. And as per McFarland's standard, 3 × 10⁴ CFE of bacterial culture was used for the study. And a control was also kept for the same, and the reading was taken at 600 nanometers. And then there is another checkerboard method called FICI, or Fractional Inhibitory Concentration Index. So the equation for FICA is MIC of the plant extract in combination by MIC of plant extract alone plus MIC of antibiotic in combination by MIC of the antibiotic alone. So by this methodology is the same as that of MIC, but if the value of the FIC, so it is shown there, sorry, okay. So if the FIC values, as per literature, if the FIC value is less or equivalent to 0.5, it indicates that the antibacterial activity is due to synergistic effect. And if the FIC value is greater than 0.5 to 4, it indicates that the antibacterial activity is indifferent. You cannot come to a conclusion. And if FIC is more than 4, it indicates that there is a negative effect called antagonism. Then we had gone for a time kill assay. It is in order to study the combined effect of time and antimicrobial agent concentration on the bacterial growth. Then we had gone for the cytotoxicity studies. The cell line which was used was human colon cancer cell line, maintained at 37 degrees Celsius, and it was dispersed in 12 well plates, and the study was done. Then the MDT assay was done. It is used to measure the mitochondrial activity, which is directly correlated to the cell viability for both attached and poorly attached cells. Okay. Then how the synergistic plant extract mixture was prepared. So the dried weight of each plant extract were weighed to 2 mg, and it was dissolved in 1 ml of water in separate tubes to get the final concentration of each plant extract as 2 mg per ml. And the antibiotic concentration used was 10 μg per ml in the ratio of 1:1. And we had also gone for the antioxidant activity because it is related. The activity of antioxidant activity is always related to this high efficiency. So we have followed the DBPH scavenging assay. Then we had also gone for the reducing power assay. The equation is absorbance of the sample by absorbance of the control into 100, minus 1 into 100. Then we had also gone for the total flavonoid content by aluminum chloride method. Then highlights in the result. Why I have put such a slide means I know you are a heterogeneous group, and I just wanted to highlight the result, and then I'll go into the details. So the plant extracts and the antibiotic individually did not show any antibacterial activity. The combined mixture of gentamicin and Colocasia esculenta had shown the highest antibacterial activity against MRSA. Activity of gentamicin was increased up to 10-fold in the synergistic mixture with Colocasia esculenta. Herbal drug interaction tested by checkerboard method and expressed as FICA index proved the synergistic effect. In time kill assay, MRSA was totally inhibited by the above synergistic mixture in 6 hours. The antioxidant study of various combinations of the synergistic mixture was proved, that is nearly 20 combinations we have done, and the MIC of the synergistic mixture was proved to be 40 mg per ml. So this is the gram staining of the culture and the reference. And this is the growth on Baird-Parker agar for selective medium for MRSA, Staphylococcus aureus. So this is a inoculum size, that is number of bacteria that is used for the study as per McFarland standard, 3 into 10⁴ colony forming unit per ml. And this is the first result, that is a zone of inhibition of plant extracts alone. This is a picture for that. That is, when we are using the plant extracts alone, the zone of inhibition is minimum. I'll show you a standard chart. In the standard chart, you can see it has to be in this range, 14, 18, 29, 22, 21, and 16. If it is only in this range, you can see that the bacteria is inhibited by the drug. So as per this, the diameter is very less, that is 2 mm, 2, 3, and 1. And it is evidenced in the pictures. This is the standard chart as per Kirby Boy method, which I had shown you before. And the synergism between solvent extract of Ixora coccinea, the first plant, and the antibiotic. So only gentamicin was proved to be, this was combined with the Ixora coccinea, and only this one was shown to be good. The solvent used was ethanol. And this is a picture for that. And next one is combination of this with the next plant, that is Mimosa Pudica. And only gentamicin proved to be good in this, and the diameter was 20 mm. And the picture for that. And next is the combination with the third plant, Colocasia esculenta, which gave the best result, because normally if it is 18 mm, you can say that it is resistant. It can be, the bacteria can be inhibited, 18 mm. But see, it is 29. So this is the best result we got, Colocasia esculenta with gentamicin combination. And it can be evidenced. And the synergistic mixture was prepared with the best result we got, that is gentamicin plus Colocasia esculenta. And the determination of MIC of the synergistic mixture. So you can see that various concentration of the mixture was right from 0 to 100. And 40, you can see it is inhibited. That is the OD value at the 0th hour, and the OD value at the 24th hour remains the same. So it is inhibited. So this is a graph showing the same. And next is FICI, or fractional inhibitory concentration index. In this, you can find the gentamicin concentration in milligram is 0.312, and this is the MIC value, minimum inhibitory concentration of the gentamicin alone. Yeah. Next is gentamicin with C. esculenta extract. You can see the value is 1.25. The graph for the same. And next is C. esculenta extract concentration alone. It is 80. Next is 30. That is aqueous extract of C. esculenta in combination with gentamicin. You can find the value same, that is inhibited. And this is the conclusion. FICA value which we got is 0.4. It shows that if it is less than 0.5, the antibacterial activity is due to synergism between gentamicin and C. esculenta. So this was a result. and the time kill assay, you can find that if you individually use gentamicin plant extracts alone, it was not killed. But when you use a synergistic mixture, it has come down. That is, time kill assay was very good. And the cytotoxicity studies, it shows that the more than 75% of the cells were viable even at higher concentration of the mixture, and assay of toxicity levels were low. An increase in concentration of the mixture has increased the toxicity level. So this is the antioxidant study result. We tried 20 combinations, combining different plant extracts and in various combinations. And this is the evaluation of antioxidant assay. You can find highest in 7. 7 means C. esculenta and Boerhaavia diffusa. This is the best result we got. And reducing power assay was highest in 7 and 16. 7 is C. esculenta and Boerhaavia diffusa. And the next one is Ixora, coccinea and C. esculenta. These are the best results we got. And the same, it is evidenced in the graph also. And this is the total flavonoid content. See, it is very high. The flavonoid content of C. esculenta was found to be very high. So that may be a reason for that. Next is phenolic content. That was also very high for Colocasia esculenta, which is evidence from the result. So the work to be done is separation of pure compounds, preparation of the synergistic drug, study of antimicrobial activity of the new drug at meta-analysis, and toxicity studies of synergistic mixture. So this is the conclusion of our work. The synergism between known antibacterial agents and bioactive plant extracts is a new concept which can be used for drug discovery of antibacterial, antioxidant, and anti-cancer activity. The synergistic mixture of gentamicin and C. esculenta showed the maximum antibacterial activity. The FICA value was less than 0.5, showing that it is a synergistic activity, and the antioxidant activity studies of various plants was also done. And these are my references. Thank you, and my acknowledgement is to my MTech student, molecular medicine student, Mr. Kumar, and also to the funding agency. Thank you.
Any questions?
Yes, I have a question. Have you tried to separate this plant extract to find a single active substance?
Yeah, actually this work was completed in May, and we are going for what is exactly happening and why the activity is increased. So we are on the, we are doing it, and it was found that, by HPLC we are trying, but I am not in a position to put it into the result. And one more thing, the permeability of the cell was increased. So that is the main reason. Whenever we use these two compounds, the permeability of the antibiotic towards the bacterial cell was found to be increased. So we are into that. We are doing some tagging studies. We are tagging with some fluorescent dyes, and we are exactly learning what is happening to this. And we can save a lot of antibiotics. It's of a major concern. Yes.
Why do you think the synergistic effect is kind of limited to one antibiotic and gentamicin? You don't see if the hypothesis, the trend is affecting the permeability, you may see more of a general effect or synergism with other antibiotics as well. But you only see this synergism with gentamicin, not with other antibiotics. So there may be, I think there may be more.
Yeah, there may be more reasons for that. But we found, actually we have done all the experiments almost 3 to 4 times, and then only we came to this. But repeatedly, only gentamicin was giving the best result. So anyway, I'm in the second phase of this research work. Maybe for the next conference, I'll be able to answer you. Sorry. Okay.
So my actual question is that
Yeah.
in terms of gentamicin resistance, what's known about the mechanism about gentamicin resistance? Because of the efflux transporter or because of some?
It is degradation only, enzymatic degradation.
So maybe that's where
Yeah.
you look.
Yeah, maybe.
Some inhibition of the enzyme by
Yeah.
Another question? I have a question. How about the mechanism of the antibiotics of the plants? Because I see only you check about the reducing.
Yeah, plant extracts alone I have done in the first slide. It is only, coming in the sensitivity level, 2 millimeter. It was very less. And it was used alone, it was not inhibited.
And how about the doses? Because in the using of antibiotics, sometimes resistant to antibiotics, while in the plant extract there's no fixed dose for the extract.
Yeah, it has to be standardized. There is no fixed mechanism for that so far. We have to, we are trying to standardize it actually.
Any questions? If no, let us give the applause to Dr. Jency Blesson.
Thank you for your patient listening.